Hi guys, this is Dr. TS, and today I'm going to be talking to you guys about the macromolecule of carbohydrates. Now, carbohydrates are really a central macromolecule because they provide us energy and structure to both our cells and to our body. Now, if you remember back to your intro lecture on macromolecules, you'll remember that all macromolecules are made up of monomers, which become a polymer. So first I want to talk about the monomer of a carbohydrate, which are called monosaccharides, which simply just means just one sugar. And these monosaccharides are going to be made up of three specific elements: carbon, oxygen, and hydrogen. And these three elements are going to be in a fixed ratio. So for every one carbon, you get 2 hydrogen and 1 oxygen. So for example, glucose is a monomer of carbohydrate. It has 6 carbons, so it has 12 hydrogens and 6 oxygens. So glucose is a monomer of, or a monosaccharide in the carbohydrate world. These monosaccharides can exist in 2 different formations. They can either be in a chain or a ring formation. So you can see they can kind of be lined up like this, or they can be in a ring formation. And both of these, both this linear and ring structure, are going to play important roles in what exactly it's doing. Now, what is the purpose? What's the function of a monosaccharide? Because we have monosaccharides in our bodies, and especially one that we're going to come back to a lot is glucose, right? This is going to be primarily where we get a lot of our energy. So monosaccharides are the primary fuel source for all of our cellular reactions. So when you're eating, a nice turkey sandwich, you're getting a lot of those carbohydrates from the bread, which will be broken down into glucose. So when we think about where is the energy in this monosaccharide? The energy is stored in the chemical bond. So for example, we have the bond here between carbon and hydrogen, and this is a covalent bond, meaning it's sharing the electrons. And those electrons are what we want. We want to get these electrons. This is the energy that we want. So in order to get those electrons, we have to break the bond. So we have to break the bond between the carbon and hydrogen, and this releases our electrons, which will be turned into energy. So energy is stored in chemical bonds. So when we have a molecule of glucose, our cells have a method of breaking that glucose apart and getting those electrons out. And you guys will learn that process in our next unit. So monosaccharides, like I said, are the monomers of carbohydrates. There are 2 types of polymers of carbohydrates that we're going to talk about. The first is simply when we just have 2 monomers together. We call this a disaccharide. Like I said, disaccharides are 2 monosaccharides that are bonded together via something we call a glycosidic linkage. So if we have 2 molecules of glucose, okay, C6H12O6 plus C6H12O6, those 2 molecules are going to come together via a dehydration. So we're going to remove water, and that will form this glycosidic linkage. Now, depending on which monomers you have as part of the disaccharide kind of changes its name. So for example, 2 glucose molecules together are called maltose. Glucose and fructose together are called sucrose. And if you read the back of a soda can or the back of a food, you might see these terms. So if you see sucrose, that means it's a glucose and fructose molecule. If you see maltose, that means 2 glucose together. What is the function of disaccharides? The primary function of disaccharides is energy storage. Remember, when we break apart these molecules into their monomers, their glucose or fructose, there is a ton of energy stored in them. Additionally, some organisms will actually transport energy as disaccharides. So some organisms might take a disaccharide of sucrose, send it to the cell where it's then further broken down into glucose and fructose. And remember, these carbon-hydrogen bonds in these monomers are what is broken to create energy, right? We get those electrons out, we get that energy out of the carbohydrate. So carbohydrates are very essential in energy storage. They have a ton of potential energy to them because they have all of these carbon-hydrogen bonds, and therefore that can be broken down and transported in order to make sure that all of the cells in our body have the efficient energy they need in order to do different cellular processes. So we have the monomer of monosaccharide, now we have disaccharide. Finally, the last polymer of carbohydrates are what we call polysaccharides. And these are going to be many, many monosaccharides all bound together. And some of the common monomers that we see in polysaccharides include alpha glucose, beta glucose, and N-acetylaminoglucose. These are just simple monosaccharides that kind of look a little bit different. So alpha glucose is going to have slightly different chemical orientation compared to beta glucose, and an N-acetylaminoglucose is another additional monomer that we see very commonly in polysaccharides. And I'll kind of mention which ones go with which polysaccharide. It's important to note that as we start to add these monomers together into a polysaccharide, that they can be straight, branched, spiraled, or kind of a combination of the two. So if we have a bunch of glucose molecules together, it can be kind of straight, it can branch off, it can become a spiral, right, where they're all connected. But essentially, polysaccharides are just a bunch of monosaccharides bound together. And they're going to be 4 specific polysaccharides that we want you to know for this class. And we're going to kind of go through each one using this table. Okay, this is a table where we talk about what organism each of these polysaccharides are found in. So is it found in plants, animals, so forth? What monomer is it made of? What the orientation of the monomers is, so is it branched, is it circular, so on and so forth. After we go through this chart, we'll talk more about the specific function of each of these polysaccharides. So let's start with the polysaccharide of starch, okay? And starch comes in 2 forms, amylose and amylopectin, and starch is found in plants. If you think of a starchy vegetable, you often think of potatoes. If you hear starch, think potatoes. Starch is made up of alpha glucose. Okay. And it exists in, like I said, 2 different forms, amylose and amylopectin. So what's really interesting is that the amylose version is not branched, but the amylopectin is. Okay, so found in plants. Then we have the polysaccharide glycogen. Glycogen is found in us and other animals. So this includes humans. Glycogen is also made of alpha glucose and is very, very branched. The next 2 are made up of different types of glucose. Cellulose is found only in plants and is made up of beta glucose and is not branched. It exists in a linear form. Additionally, chitin. Chitin is a really interesting polysaccharide that we see in organisms such as like arthropods. It's found in like the exoskeleton of insects. This is made up of that really long N-acetyl-D-glucosamine. Say that 3 times fast. That is also not branched. That also exists in a linear form. So we have these 4 different types of polysaccharides with starch, glycogen, cellulose, and chitin. Starch and glycogen are both made of alpha glucose and are highly branched in some cases. One's found in plant and animals. Cellulose and chitin are not branched, and they have very specific types of monomers like beta glucose and N-acetyl-D-glucosamine. Now, how does this relate to its function? So let's first talk about the two that are made of alpha glucose, starch and glycogen. The function of both of these is going to be the same. It is primarily energy storage. This is why we see a heavily branched polysaccharide function, right? Lots and lots of branches of these can fit these molecules of glucose all together and store lots and lots of energy. Now, we eat a lot of starches. I love a potato. We also get a lot of energy in our glycogen. So we would say these are digestible. Yes, we are able to digest and eat starch and glycogen. We can break them down into their subsequent monomers of alpha glucose. However, cellulose and chitin, because they are made up of, remember this is beta glucose, and then this is N-D-glucosamine, they are not digestible, okay? They are not able to be digested by our system. We do not have enzymes in our body that are able to break this down. And so what's really interesting is that in plants, cellulose and chitin and the exoskeleton are primarily providing structure. So far we've talked a lot about how carbohydrates are really important in energy storage. They are also really important in structure, specifically like what we see in plants in the exoskeleton of insects with cellulose and chitin. So if we think about the structure of these, right, we talked about how starch and glycogen are highly branched. They are made up of alpha glucose. They are digestible. So the structure is going to dictate the function. This is going to be a theme that we come back to a lot. What something looks like often tells us what the function of it is. On the other hand, cellulose and chitin, right, which are both linear, non-branched, they provide a lot of structure to things, and our body isn't able to digest these because we don't have enzymes for them. All right, so this kind of covers our lecture on carbohydrates that exist as monomers and disaccharides, monosaccharides, polysaccharides. Thank you, guys.